All right, so welcome to today's end-of-year roundup for ENIGMA. We really appreciate you all joining. I know it's been a stressful year, and I certainly appreciate all of your leadership and guidance and sheer stamina in getting through a year that's been very stressful for us all. So the agenda for today, we'll give a quick update on the overall structure of ENIGMA, highlighting some new working groups initiatives. There's updates on grants pending and awarded, some new methods and toolboxes for us all to use, and really just a review of recent highlights of manuscripts and projects, conferences and meetings, and some announcements at the end. So just as an overview of the last year, there'll be some positive news in all of this. Sophia's updated this map, so there are now an extraordinary range of projects and working groups that you see represented in the key, with over 2,000 scientists co-authoring papers participating in ENIGMA. The newest edition, and thanks to Charalambos in Cyprus, who is the 46th country to join, and he's participating in Lee's ENIGMA Stroke Group. So thanks so much to everyone who's made this possible. There is a newsletter. In fact, if you download the PDF link in the email that Sophia sent 1 hour ago, you'll be able to click on all these links. And if you have colleagues that are interested in subscribing, they can subscribe. There's also a range of booklets and digests of conference abstracts, posters of various kinds that you can find following the link on the website. So here's an updated graphic. So we now have 51 working groups in ENIGMA, many focusing on psychiatry and neurology, others looking at neurodevelopmental conditions, and also some new groups that are highlighted here in methods and clinical areas. Just to say a little bit about the organization of these different groups, TBI, which you see, the brain injury group highlighted here, has a number of subgroups that all have their own calls, and papers will highlight some of those in a minute. But I do want to call out 3 of the groups that are new that perhaps might be of interest to all of us. So the first one is led by Alison Nugent at the NIMH. She's an expert on the clinical and technical aspects of MEG, and she's, with her colleagues Anna Namist and Jeff Stout, really done an absolutely fantastic job of recruiting groups from around the world to cooperate on an analysis of MEG. I don't know, Alison, I know you're out there. Do you have a quick update on what is going on, maybe briefly just to tell us about what's been going on with the startup of the MEG group?
Sure. So we're kind of at the point where we've contacted a bunch of groups around the world that are interested, and we have about 30 to 40 that are planning on signing on. We think we're still working on that. And we're just moving to the point where we'll start getting data from each group. We're thinking of doing this in more of a meta-analysis framework rather than meta-analysis, because we do have the resources at the NIH to be able to analyze the data. And I think the thing that we'll really be able to offer is the ability to use source localized electrophysiological data. And our initial project won't involve genetics, just because we wanted to be able to include the maximum number of MEG labs around the world. So initially we'll be looking at things like power spectral density and peak frequency within bands, and correlating those with different demographic variables and participant characteristics. And we also think that in kind of our initial data pool, we'll have several patient groups that will be represented enough that we can have some secondary analysis, probably on TBI and ADHD and epilepsy, and autism spectrum disorder. That's another one we'll probably have a lot from.
Thanks, Alison. I really appreciate your leadership and bringing that group together. It's a wonderful, wonderful initiative. You also want to highlight the ENIGMA-Environment group. So you'll see Lauren Salmon, Megan Herting, and JC Chen, who are here in Los Angeles at USC. They have set up a group that will help you do geocoding of your scan and cohort data, and then link it to environmental information, either on pollution, urban density, all sorts of other climate-related and environmental data. Exciting new effort. If you want to join, do contact them. There's a poll on what sorts of data you have. You do not have to have geocoded your data to take part. They'll be holding workshops to help you do so. So this has already been launched with over 19,000 individual scans. And we're really excited about this group also. Simon has a new initiative on language and the correlates of language in the brain. I don't know if, Simon, you want to give a very brief update on this initiative?
Yeah, hi, thanks, Paul. Very briefly, just to say, so we're at the moment just exploring the potential of starting this project where we want to look at the consequences of, or the correlates rather, of speaking more than one language for brain development and brain function. And we want to relate that to the ages at which people started learning those languages. And you don't need genetic data at this point. We're just gathering information on who has data sets with the number of languages that people speak, what languages they speak, how fluent they are, and so on. And so far, there's a link here you can see. Please, if you have this data, sign up. And we so far have about a few thousand participants, but we'd obviously like to get to a bigger sample size. And it's possible that we could develop this into a new working group on language more broadly. So if you're interested in that, please get in contact.
Very, very exciting. Thanks so much, Simon, for your leadership with that very, very cool topic. There's also a new initiative led by Thomas Hayek, Niels Opel, Scott Mackey and their colleagues, who each individually had found a moderating effect of body mass index, or BMI, on the effects of the disorders they study on the brain. So they are going to be in touch soon, but anyone interested in the influence of BMI on the brain, either itself or its moderating effect on the different disorders, should definitely get in contact with them, and there'll be more information on a Mendelian randomization study that they're about to begin very soon. There's also a special interest group focusing on machine learning, led by Lee Jolens and Johanna Bayer, Philip Seaman, Laura Hahn, and Leon Schmal, that's had a number of meetings. There's actually a recording here of a very interesting technical discussion on harmonization of data across sites and some of the more advanced methods, combat, hierarchical Bayes regression, and deep learning for doing those things. There's actually a question for the chairs, and I think, Sophia, we're going to do a poll. I don't know whether this is possible, that Philip had asked the question for the machine learning projects. What kinds of predictions are you making? What kinds of variables are you trying to predict from the imaging data? I don't know if it's possible to do a poll right now, but we'll have a go.
Yeah, so we don't have a poll for this question, but if all of the chairs want to type out in the chat, we'll take a look at that. We have a poll question coming up later in the call.
That's perfect. And so, just as a note, Joaquim Radoa led a project testing combat as a harmonization method, and a new and improved version that he's written that handles missing data, among other things. So do have a look at the paper he published in Neuroimage with a number of colleagues there as a possible approach for all of us. There are several discussion groups that have been formed by the ENIGMA-Epilepsy working group on genetics, new methods, and cognitive data harmonization. This grew from a workshop, very interesting workshop, that Sanjay Sisodi and Carrie McDonald held in August. If you'd like to watch it, the recordings are at the link there. It tackles many issues in harmonizing imaging, genetic, and cognitive data. And you're very welcome to join their ongoing working group discussions. Some new grants. A big congratulations are due to Li Lu for her stroke recovery R01 from NINDS. NINDS is also supporting a new initiative led by Frank Hillary, Emily Dennis, Lisa Wilde, and David Tate on tool development for understanding brain injury. A new R01 from the NIH was awarded on brain injury also to Kari Esipenko and colleagues. In the running for funding, the ENIGMA World Aging Center, which supports the lifespan, plasticity, and epigenetics groups, scored a 6th percentile. So that looks like it's in the running for funding. The Parkinson's disease R01 also got a relatively good score. And pending, there are grants on epilepsy, anxiety, a grant that was submitted to the Fogarty International Program by Ninka Gronewold, and also a cross-disorder grant by Peter Kochanov, and Perry-Pasco's Tourette syndrome group are all pending at the NIH at the moment. And a couple that you'll see there are in revision for being resubmitted. Here are the funded grants that are supporting different activities in ENIGMA. You'll see a broad spectrum from addiction, suicidal thoughts and behavior, and PTSD, right through to more technical grants on genetics and task-related functional MRI. There's a project called ENIGMA CoinStack, which is testing distributed computation in Vince Calhoun's CoinStack method as a way to operate on data from multiple sites. And also a new grant to Ian Harding from the NHMRC in Australia, which has led to some really fantastic developments in ENIGMA ataxia. So I just wanted to highlight a few different developments. The team here at USC, including Neda Jahanshad and her student Yad Bagari, have been creating some software for navigating and QCing the data that we're all going through. The tools for DTI are being upgraded by Dr. Jahanshad and her student Elizabeth Haddad. And also there are some wrapper scripts for many of the popular processing pipelines that have been developed by Li Lu and her colleagues for the stroke recovery group. Of course, these are of very general interest and utility to all the working groups, so do check out the link that you see there. There's also some very good updates from the task-based fMRI working group. And I know, Ilya, you're on the line. I wonder if you could give us a quick update on the status of this project.
Yes, hello, I'm here. I think I will leave this slide to Henrik to give an update on, because he has some new information. And the next slide is on the pipeline, and I can say a few words about that.
Great.
Yes, hello. So as we were very unsatisfied with the imaging genetics and functional MRI, we said we want to know how this goes, if we have really this large cohorts, and started this ENIGMA task-based fMRI working group. And it will have a kickoff meeting in January. You see the link here. And we just started off because we have 9 labs worldwide, which already provided a letter of intent. And the idea is to collect types of paradigms. If we want to start with emotional phase processing, like the Hariri task, reward processing, like the monetary incentive delay task, and working memory. And we have already in the letter of intent nearly 10,000 people in those things. And for example, like Ahmed Hariri from Duke University is on board, and Alex Ronita from Monash University, and Steve Smith from UK Biobank, and some others. So yes, if you're interested to take part, this would be interesting if you have these types of paradigms in larger groups, maybe healthy subjects or patients, and you can just mail us. You will have there's the contact, the enigma@charité mail, or you can come to the kickoff meeting, and we will be very happy to do this. And we have, in order to be able to do meta-analysis, have worked the last year on pipeline, which allows distributed analysis. So you don't have to share data, you just share results. And Ilya will now shortly explain how this works. Thank you.
Thanks, Henrik. And Ilya, you have some updates on resting state also.
Well, not necessarily on resting state, more an update on fMRI data analysis in general. So the past 2 general meetings, I already presented a bit on ENIGMA HALF-PIPE. So this is our developed pipeline. We worked hard over the past 2 years to make it possible to do harmonized analysis of all kinds of fMRI data, so either task-based or resting state, from raw data pretty much to group level statistics. So this is a short summary of what our pipeline can do. It's built around fMRI prep, and we added layers of quality control and statistics on top of this. So I will also post now the link again in the chat, so you could have a look at our GitHub repository, and there's also information on how to use it. So if you want, we are currently in our final beta version, we think. You can download it, play around with it, and see if it works for you. And we also value any input you might have on this. So this will be central to our endeavors in task-based fMRI, and also in the resting state fMRI working groups to arrive at a GWAS, which will be announced later on task-based imaging and resting state imaging. And maybe one more thing to add here is that for now we have done this under our German grant, but recently Emily Dennis and Frank Hillary have been awarded an R61 grant. I'm also part of this as a consultant on the pipeline, and this is actually served to further develop and maintain ENIGMA half-pipe, so also that it will in the future work with, for example, distributed combat and CoinStack. So this will be in the next 3-4 years, I guess, migrating to this grant.
Thanks so much, Ilya. And just drawing attention here to Carissa Weiss's work as part of Raj Mori and Mark Lowe's PTSD working group that successfully run the half-pipe method across a large range of cohorts with fMRI and PTSD. So this is becoming a really good suite of tools and a great roadmap for us all to test out. So thanks, Ilya, and Leah, Suzanne, and Henrik for doing that. I do want to mention Matthew Kempton and Tony James's VBM tool, which is now being tested in early onset psychosis, OCD, and PTSD. And Matthew was on the call 5 hours ago about OCD talking about this. Do check it out. The manual is extremely thorough, and we appreciate all of the work that Matthew, you and Tony have done on this. There's also, under Ian Harding's leadership, he leads the ENIGMA Ataxia group, a suite of tools for the voxel-based and segmentation analysis of the cerebellum. So he's packaged in a very, very nice set of tools. Some of them are Docker tools that can be run remotely. A number of popular methods for cerebellar analysis. Also, these are being implemented in OCD by Janardhan Narayanaswamy and Srinivas Balachandra, who presented this morning on the OCD call. And this presents a really useful approach for getting at cerebellar anatomy, which is of great interest to a lot of us. Also want to highlight a Toolbox that was developed by Sarah Larivière and Boris Bernhardt at Montreal Neurological Institute. They conducted a study of structural covariance in epilepsy, but in the process of doing so, developed some beautiful tools for analyzing all sorts of cortical data that will be valuable across ENIGMA. So they have tools for graph theoretic analysis of cortical measures. There are tools to correlate your cortical maps with histology and omics data. And I would encourage you to check out this Toolbox link there at the bottom. I mean, there's some really wonderful tools that I think all of you will find very valuable. So thanks to Sarah and Boris for developing these. There's also a press release if you're interested in a very nice new study in epilepsy of hubs and epicenters of the cortical network in epilepsy. There's also a machine learning toolbox developed by Hugh Garavan's lab, Sage Han in particular, who was lead author on a terrific tool where machine learning was applied in the addiction group. One of the nice things about this toolbox is it copes with very large data imbalances. You can even include cohorts of patients only, or for that matter, controls only. And there are a variety of methods to handle not just missing data, but also to run many complementary and popular machine learning analyses in tandem. So do check out Hugh and Sage's links here. Okay, so quick overview of published manuscripts. There's just been an extraordinary productivity of over 50 published manuscripts in 2020 alone, which is a real testament to the leadership and staying power of many of the groups. Sophia's very kindly compiled this with links to all of them. So if you have the PDF, you can have a look at all of these. Specifically, there was a special issue that Gary Egan at Human Brain Mapping invited us to create with about 35 papers on technical, clinical, and logistic aspects of ENIGMA, including many new empirical papers. Now, this will appear officially in March, although many of these papers that are highlighted in green have already been accepted and appeared online. So do check out this list of papers. The ones in white are still pending, and they're hopefully going to be accepted very, very soon. But I think this will be a really fantastic document to look through all of the things that are going on across ENIGMA. Also, Emily Dennis, in partnership with the Brain Imaging and Behavior Journal, organized for a special issue in which many of the working groups that are part of her brain injury working group for ENIGMA have described the challenges they encounter, their roadmap to solve them. And many of the particular things that occur in studying brain injury in military, sports, and adult and pediatric populations that you see here. So I strongly encourage you to check this out. The links are there to the papers for the ones that are already accepted, and congrats to Emily for making this a real success. Going on to more specific areas of analysis, many of the groups have completed their cortical analyses. You'll see the ENIGMA t-shirt, which used to have all of these maps on, now needs to be expanded to include several others. There's a paper newly out by Xin Wang in Raj Mori and Mark Logue's PTSD working group, in which they also did voxel-based cortical analysis. There's a new cortical analysis by Gemma and Andre's schizotypy working group that's been submitted. And ongoing is Stefan and colleagues' antisocial behavior cortical analysis project. Now, this has also paved the way to a vast and gigantic analysis of cross-disorder data. And so Elena Pozzi, Chris Ching, Theo Van Elp, and Alex Muir have been working on an extraordinary transdiagnostic analysis of 119 cohorts across the working groups listed here. And they've been able to model for the first time symptom subdomains and other data that's been collected across these working groups in really quite a remarkable analysis, whose preliminary data you can see here. So congrats to them for that project. Many of the ENIGMA working groups have begun with studies of subcortical shape. All of the published ones you'll find listed here. I do want to call out on the right here some shape analyses that are newly published. Chris Ching and colleagues have studied shape in 22q. There's Tiffany Ho and Boris Gutman's study of major depression, and also Yan Shai and Hugh Garavan and Patricia Conrad's study of substance dependence with shape analysis methods. And on the bottom line there, you can see there's a large number of groups doing shape analysis in a variety of different conditions. So one of these is the first paper by the stroke recovery group led by Li Lu. Just wanted to highlight this because this is the first paper that they've done. Do check it out on bioRxiv. It's a really interesting study of the effects of lesions on post-stroke behavior, meta-analyzed across 28 cohorts. Also, large number of efforts looking at fine scale anatomy, both vertex-based analysis of subcortical structures and cortical structures. You'll see two new ones, Lei Wang, Boris Gutman, and the schizophrenia working group, mapping the effects of schizophrenia across the basal ganglia and other subcortical structures. And a companion paper by Matthias Kirchner and Gemma Andre in the schizotypy working group, where they're beginning to look at subcortical volumes in fine scale in that disorder also. Now, it's been a great year for ENIGMA for diffusion. And so the diffusion MRI studies that you see here tabulated are all published. You'll see a digest of the number of cohorts and the age range and kinds of data with links to all of those. Ongoing at the bottom, you'll see the groups that are currently doing diffusion studies and the leaders of those. So this has been a tremendous success. Just if we can highlight one in particular, Sean Hatton and colleagues, as part of the ENIGMA working group, really did an absolutely remarkable job of analyzing cross-cohort epilepsy data with different types of correction. You can see combat applied here as an illustration. So for those of you that are embarking on a DTI analysis or want to plan one, I'd very much recommend Sean's paper as a sort of roadmap and guide to the types of things that could be done. In genetics, and the crown of this year's work has been the paper in Science led by Katrina Grasby and Neda Jahanshad, that has also been led by Sarah Medlin, Jason Stein, Barbara Franke and Janita Bralton and their teams in Brisbane, Nijmegen, North Carolina and Los Angeles. Sarah was interested in getting pictures from people that you can see pictured here with the paper that came out. And if you look there on the top, there's the printout of the paper that was very meticulously proofed. So thanks so much again for that remarkable study that discovered hundreds of genetic loci that are associated with cortical surface area and thickness. And these projects are also ongoing and being topped up. A shout out to the ENIGMA Evolution group, Jason Stein and Amanda Tilo with Simon Fisher and many of their colleagues, just published a paper on the evolutionary relevance of some of the cortical loci that have been discovered in their evolutionary timing and emergence. There's a wonderful video that you can see linked to the bottom where Jason explains how to study evolutionary aspects of cortical anatomy using genetic data. His lab also, if you look at the paper in the middle, has just published really interesting study of how discoverable, how easy is it to find genetic loci that influence brain traits, and for that matter, psychiatric and other behavioral traits. And they give us a lot of insight into the polygenicity of these different conditions. Also, Sarah and others have completed an overview of 10 years of work by the Genetics Working Group that just came out online in Human Brain Mapping, so do check out that study. There are also, next year, planned large-scale genome-wide association studies of, first of all, diffusion imaging and white matter. This is being led by Neda Jahanshad, Barbara Franke, Simon Fisher, many other groups. 128 data sets are being included. They'll be in touch in January 2021. If you haven't received any information on this, don't worry, they'll be in contact very soon to make sure that all those interested and able to do that can take part. Another major direction will be Ian Harding working with Sarah Medlin's group to analyze the cerebellum. They'll be providing you with protocols and scripts and information on how to take part in that. And then there's also many additional projects that apply more advanced multivariate analyses to look at cortical genetics. A project led by Vince Calhoun and Jess Turner looking at the ApoE2 Alzheimer's protective genotype that cohorts will be able to opt into if they like. And then there's also future plans for resting state and task-based GWAS that I think Henrik and Ilya alluded to earlier on. Just a reminder that Sarah Medlin does have funding to genotype your cohorts. This has been a really great success with around 6,000 samples being genotyped. If you have phenotypic data, MRI scan data, and DNA that you do not have genotyping for just yet, ideally in a cohort of over 100 people, do reach out to Sarah and she'll try to set that up for you. And also just to draw attention to the ongoing polygenic risk projects looking at the effects of polygenic risk for different disorders on predicting brain morphometry. You'll see a number of groups that are taking part or leading PRS studies at the bottom there. Another real success this year was a project led by Yash Patel and Thomas Paus, where they were looking at the project called Virtual Histology. They were looking at the gene expression for various cell types across the cortex and how that relates to the patterns of deficits and abnormalities that we have been seeing in a range of psychiatric and neurodevelopmental disorders. Now, this project is truly remarkable. It gives some hints as to the cellular correlates of some of the alterations we see in different psychiatric and neurodevelopmental conditions. They're also leading an extension of this project into neurology, including Parkinson's disease. So if you lead a working group that's finishing a cortical project, do get in touch with Yash and Tomas, and they will provide some really nice scripts to your group members to relate their observations to histologic markers. Very, very interesting line of work. We also wanted to give a quick update on machine learning, and particularly Brain Age, which is one example of machine learning across ENIGMA. So already published, there are a number of papers, Scott Mackey and the Addiction Group, Abraham Nunez and Thomas Hayek, the Bipolar Working Group, and several others have either before this year or this year published machine learning analyses, either for diagnostic classification or prediction in the cohorts. Newly submitted, congrats to Zeke Leichert and Leo Bonilla for their deep learning study of epilepsy, where they contrasted different classical and deep learning methods for classification of epilepsy patients versus controls. And then also a very elegant study by Heather Bouchard and Raj Mori using non-negative matrix factorization as a multivariate distillation of DTI measures in a machine learning study also. Now here is a tabulation. I thought that we would all find this interesting, of all of the ongoing projects across ENIGMA, at least that we know of. And you'll see certain themes. And so some of them have certain clinical and neuroscience goals. On a technical level, there are certain commonalities in the methods. whether they use classical or deep learning methods, whether they're looking at biotyping or clustering. So again, I'd encourage each of the groups listed here perhaps to reach out to some of the others, because I think collectively, there's also the group led by Lee Jolens and Johanna Weyer. We can all discuss our findings, and this is a very exciting area I think that ENIGMA can contribute to, given the range and diversity of the cohorts for training and testing machine learning methods. On Brain Age in particular, James Cole has ably led a very nice working group on Brain Age in ENIGMA, where many of the working groups that you see listed on the right have been testing different Brain Age estimation methods. Just a shout out to Laura Hahn and her colleagues and Lian Schmal, who've published an algorithm that they trained on ENIGMA's major depression working group data. This was then tested by Lisa Eiler and colleagues on data from the bipolar disorder working group. One very nice thing about this is that Tim Hahn is hosting the Brain Age algorithm on his website, photon.io. And so if you follow the link at the top, you can actually upload, if you want, CSV files of free suffer measures that have been anonymized and get back estimates of brain age. So this is obviously going to lead to a great deal of interest. And I think you'll hear more in the new year from James and the genetics working group and Esther Walton on a GWAS that is planned of brain age measures. So in an effort to sort of coordinate and see connections between activities across working groups, we made a list of the different analytic methods that have been applied to the imaging data that is being processed. And you'll see them listed here. So the structural covariance analysis, I mentioned the very nice work by Sarah Lariviere and Boris Bernhardt. There are many other wonderful projects. Jonathan Ottino has just finished a paper on structural covariance in the addiction working group. There are many others that are underway. Adeline Sun and Raj have a very nice analysis underway. Other themes are looking at medication effects on brain structure. You'll see many of them listed. There's a CBT project that's being led in the OCD working group. Also a number of efforts to look at early life factors that influence the expression of psychiatric illness. Here you'll see a couple. And also several working groups are beginning to look at longitudinal MRI data. So following on the heels of Rachel Brower's plasticity working group, which did an extraordinary GWAS of brain change in serial MRI in 10,000 people. It was actually a remarkable study. Some of the other working groups that have longitudinal data are running analyses. If you are listed here or your members are listed here, do encourage them to reach out to the other groups tackling the same questions, because I think there's tremendous potential for synergy across here. I won't list them all, but as you can see, there are many thematic commonalities among the projects in different working groups. Again, thanks for putting up with the online format. I think the all chairs, all hands meeting in the summer was a great success. We'll certainly do it again. If you missed it, there are recordings of the entire meeting at the link that you see below. There are many topics that if you have students and researchers who are new to ENIGMA or to multi-site analysis, they can go and have a look at those and really get up to speed with some experts in the field who've led these different analyses. So the all hands meeting next year is likely to be virtual. I think this is the point where we're going to do a poll. So I don't know if Sophia, you might want to give us some advice on what to do here. I was very interested in whether or not an all hands meeting should be in conjunction with a formal meeting such as OHBM or SOBP. In some sense, it doesn't matter, although you should be take care not to clash with other activities that people have committed to do. Also, the TBI group in ENIGMA are planning a virtual conference on TBI, looking at dedicated funding that you can obtain to run meetings. I do want to say, a note of thanks to the epilepsy group. They had a very successful morning symposium that lasted about 4 hours, in which the groups really could brainstorm in greater depth about new directions and new methods that they were interested in. So I think, Sophia, is there a poll, or is this something where people...
Coming up. So in a few slides, this is also just, if you have any feedback on, because I know everyone's on Zoom all the time, or on their screen. So if you have any feedback on the day, the format, if you feel like you'll have a fresher mind before all the conferences begin, or if you like it when it's between all the conferences. If you have generally any feedback, feel free to email us or post it in the chat. We'll be saving the chat and going over everything afterwards.
That's wonderful. Thanks so much, Sophia. And just as a sort of highlight, so before the pandemic hit in March or so, there were actually a number of conferences. There's some here in Denver and Hawaii and all sorts of places we wish we were at the moment. Just a big word of thanks to all of you who organized and took part in symposia that presented ENIGMA among other topics. I know for next year, SOVP and OHBM will be good targets, but also the meetings on particular disorders you'll see listed here are very, very good venues to introduce people to the work that you and your members are doing. So thanks so much for that. Just as a note, many of you know this, but there are conference deadlines coming up where many of the projects that you're leading or taking part in can be presented. OHBM has a deadline of February 1st, SOVP February 11th for late rating abstracts, and there's a couple of new abstracts just listed here, including a very nice project led by Leyland Balsi and Dara Cannon on meta-analysis of white matter connectivity with diffusion. It's one of the first projects that's looked at that, so that was very interesting. And if we've missed any, do send them to Sophia to make sure that we've mentioned your project here also. So just a number of announcements. This one is very interesting. So we've been promising for some time to have a webinar series for ENIGMA, both to present updates on recent papers, to talk about methods that might be of interest to many working groups, and also just to brainstorm about future options and directions. So I think we learned how to do this from Megan and Lauren's ENIGMA-Environment working group calls. They had a number of polls. So if you'd like to fill out this poll here that you see popping up on your screen, there's a number of questions, about what times of day. In fact, I'll just do it now. I would like to do this in the early morning, and I'm absolutely fine with meeting on all of these different days. So if you return your responses to that, hopefully it worked, and we'll try and organize that to be convenient. Also, here's a number of videos and updates. So very often we're asked by new members, can you tell us a little bit about ENIGMA? What are the goals? What types of things can I be involved in? There's a number of new videos here that are interesting to share with your members and students and colleagues. There's an interview series that Sophia actually filmed. And so when we were in person, many of you will recall being interviewed. Some of those interviews are still being produced. So there's some coming soon on stroke. There's Rebecca Santelli's group on infant development and genetics called Origins, and an interview with Sylvain de Rivière on epigenetics. The new one, you can see Jason talking about evolution here, but do click on the whole series. It has a whole range of very interesting things. Jess Turner gave a terrific talk. I really enjoyed this talk. It's an hour-long talk on ENIGMA and CoinStack and some of the philosophy and successes and challenges in doing multi-site data analysis. That also is on YouTube. You can also see me and my cat giving a talk about ENIGMA if you click that link there. And then again, Boris and Carrie and many others hosted at McGill a workshop that I found fascinating, clinically and technically, on connectomics and epilepsy. And you can see the recording. It's on Vimeo, and so you can look at the lectures that were given at that. Okay, so this is sort of interesting opportunity. I don't know, Chris, if you're on the line, if you want to say a couple of words about this one.
Yeah, sure, Paul. So we have a new opportunity to maybe publish on some of the ENIGMA standardized processing pipelines, protocols, toolboxes. So the editors of this journal, JOVE, which is the Journal of Visualized Experiments, approached us about maybe putting together an ENIGMA methods special collection. So this is a peer-reviewed online journal that publishes papers and videos of scientific protocols. The goal is to improve replication and reproducibility of experiments, The submissions include basically writing up a short paper on a method or a protocol, similar to what we do with these ENIGMA standardized protocols, with some simple applications or examples. These can include previously published data or results. And then once the paper is sort of peer reviewed and accepted, they have a production team that puts together these really nice professional videos that summarize the method or protocol that goes along with the paper. And obviously all of this is citable. And so, you know, we've had a number of ENIGMA working group members have published with the JOVE folks. I'd encourage people to take a look at, you know, this recent article from Joaquim Radua and Edward Vieta's group, especially to take a look at the video quality that comes with sort of the submission. And so, yeah, basically we're just looking to see who might be interested in potentially submitting on one of these sort of protocols or toolboxes people have been putting together. And we'll be following up with people early next year about potentially putting together a special collection. And the last thing I'll mention is, you know, once you film these videos, you're free to put them on your website and distribute them as you like. But it's an interesting platform, and yeah, let us know if you're interested.
Thanks, Chris. So also, just as a reminder, Sophia has been collating a large number of resources for chairs of working groups. So if you look at these links here, there are all sorts of documents and templates for welcoming new members, answering frequently asked questions. You know, many of us have been tackling, you know, different models for authorship. I mean, there are certainly papers where there are primary and secondary contributions and how to sort of handle authorship in these large-scale studies. There are template forms that you're free to adapt and modify for your own group's purposes on secondary proposals, and also MOUs and other sorts of things. The other quick update is that if you have a newer working group and you would like Sophia to update the ENIGMA website with some information on the members and goals, do send that to her. And I think Sophia has a note on if you want help chasing authors, you can also enlist her help, which is very valuable. So, also a note of thanks to Emily Clark-Rubright. So she has been the project coordinator for the ENIGMA PGC PTSD working group for the past 3.5 years. I don't know, Raj, if you just want to say a few words. I'm tremendously appreciative of Emily's work and learned a lot from what she presented on cognitive data harmonization. I don't know if, Raj, you just want to say a few words of thanks to Emily. Oh, maybe Raj isn't on. Sorry to put you on the spot there, but we sure appreciate everything that you've done, Emily, and good luck for your next endeavors.
Thank you so much.
Sure. Thanks, Emily. So that's about it. And miraculously, we're done in 45 minutes. But I just want to say, you know, ENIGMA would not be where it is if it wasn't for the leadership and insight and sheer vision of all of you. I think the successes that we've talked about have relied tremendously on all of your hard work, all of your planning, all of the intellectual sagacity that comes from managing large-scale projects. Really, we do hope that next year is a little easier on all of us. I think the successes that we've mentioned today, you know, many promising future directions. And so just once again, we owe you all a great debt of gratitude. Thank you so much for all of your participation and leadership and staying power. So that concludes, I would like to say, the final teleconference of the year for ENIGMA, but I know there's actually 3 more. And so if you're missing us, you can join the Schizophrenia Working Group call at 6 a.m. Pacific time tomorrow. And there's also an EEG working group call on Monday. But with that, I will say thank you. And thank you to the team here in Los Angeles, notably Sophia Tomopoulos, for making these slides and keeping us on track with everything. And good wishes for the holiday, and all the best for a much better 2021. So thanks very much. Because I did the whole call far too fast, and it should have been 15 minutes longer. So go ahead, do
15 minutes. So thank you, Paul. Thank you, Sophia. So what I wanted to say is that I'm moving from KCL to Charité and Fudan to do population neuroscience. But the reason, the real important part about this is that at Fudan, I have about 5 professorial positions to fill. So on the full professor level and on the associate professor level. Now these professorial positions, can be full-time, but they do not have to be full-time. So they are not necessarily, so to speak, in conflict, with your current work. In fact, dual affiliations would be appreciated, and also part-time positions would be appreciated. So I was just going to sort of raise that, and perhaps Sophia and I can have a chat afterwards, or an email exchange afterwards, to sort of circulate a little bit more information about that. So if you know anybody who's interested, I'd much appreciate to hear from you. And I can say that Fudan is very nicely equipped, so to speak. They have a 7 Tesla, they have 3 Tesla connectome, they have a 11.4 animal Tesla, they're a pet. So there is at least a lot of gear there, and there is a lot of enthusiasm there. And I think it would be nice to build something up together, of course, with the Charité link. So if you know of somebody, if you're interested, please let me know. Now so these are the sort of 2 sets of announcement that I wanted to make, and thank you very much.
Thanks, Gunther. As always, it's great to see you, and I always admire your pioneering spirit in doing all of these different projects that you lead. So thanks so much. Are there any? Sorry, go ahead. Yes.
This is Raj, and my mic wasn't working when you asked me to speak earlier, but I just did want to say some words about Emily Clark, who's been in my lab for 3.5 years and leading the ENIGMA PTSD work group, and just done a fantastic job with corralling imaging data as well as genetics data, working jointly with ENIGMA PTSD and PGC PTSD, and I think kind of like set the standard for kind of organizing working group efforts, and did a lot of kind of groundbreaking organizational things. So I just really thankful for her efforts, and thanks for recognizing her, Paul. I appreciate it.
Thanks, Raj, and thanks to Emily too. So are there any other parting remarks? There were some questions about whether the slides for this are available. They certainly are. So if you check your email for a link from Sophia, the PDF is terrific because it has all the links to all of the extended papers and documents that we've talked about. So thanks so much. Does anyone else have any final thoughts or comments before we go off into the new year? All right, jolly good. Well, all the best from Los Angeles, and have a good evening and rest of your day. Take care.